Hi everyone, it's Ms. Lopez. In this video, we are going to continue with our notes on chapter 2. This time we're going to focus on carbohydrates, the first of the macromolecules we're going to use and discuss in class. Before we get started, I wanted to talk to you guys about functional groups. So functional groups are small groups of atoms with specific chemical properties, and there's some common functional groups we're going to be talking about. Functional groups confer these properties to the larger molecules. So sometimes if a functional group is polar, a larger molecule that may not be polar will have a polar section on it because of a functional group. And keep in mind that one biological molecule may contain many functional groups. Now, a functional group that you all are already familiar with is the hydroxyl group, which is OH. And so typically what you're going to see is a molecule of some kind. Let's say this is several molecules joined together. This is just a generic representation. But what you're going to see with these functional groups, especially in the case of the hydroxyl group, what you're going to see is an additional line coming off of your molecule, and then the subgroup is written in. Okay, so OH. This molecule has one functional group. One of the things that I do, you'll see in many drawings, let's say that the functional group was on this side over here. I want students to keep in mind or remember that what they're looking at are bonded atoms. And in some cases, like hydrogen is the most obvious one I can think of off the top of my head, it can only form one bond with another molecule. So there's only one shared electron pair here between the oxygen and the hydrogen. So even though what you may see is a functional group, the same exact functional group, I may write it different ways to show what is bonding to what. So on this side over here, I wrote it as OH, but on this side over here, I'm going to write it as HO, because I want students to keep in mind that this functional group is attached at the oxygen, not the hydrogen. They're the exact same functional group. I'm just trying to show or illustrate or model what is bonding to the larger molecule or the macromolecule in this case. So even though it's written as HO on this side and OH on this side, I'm trying to represent the same functional group. And this one is the hydroxyl group, which you guys learned from your properties of water vocabulary. Okay, so now that that is kind of cleared up a little bit, let's talk about carbohydrates. So macromolecules are polymers made by covalent bonds of the smaller molecules called monomers. And what they're showing you here is kind of the makeup of living organisms. Let's say that this is a human. You can see that they are mostly water. There are a small percentages that are ions and small molecules, but it's these macromolecules that make up a large portion of the organism as well. Not as much as water, but still a good chunk. And when you take the macromolecules and you break that down into the individual types, you'll see that proteins make up a large portion of those macromolecule groups. Nucleic acids make up next, then the carbohydrates, and then finally a small portion that make up the lipids for that group of macromolecules. So you had the 20 macromolecules that you learned from your vocabulary. You learned about proteins, which are made of amino acids, carbohydrates, which are made of monosaccharides, nucleic acids, which are made of nucleotides, and then lipids, which there's no true monomer, but if we had to say monomer, we would say triglycerides. Although for lipids, there's no true monomer in the same way that we had with the other ones. Keep in mind, or one of the things I want to make sure that you guys know is that something that gets confused often with students is this term amino acid. Many times they want to tie amino acids to nucleic acids. I'm not sure why. I have a hypothesis, and that is the word acid and the word acid, maybe they think that the two go together, but they don't. So just kind of make special extra note of that, that amino acids are not the building blocks of nucleic acids. Nucleic acids are made of nucleotides. Okay, so polymers are formed and broken apart involving reactions with water. So you have condensation, which is the removal of water to bring monomers together. And then you have hydrolysis, which is the addition of water, breaking the polymer into individual monomers. Sometimes you will see this, and you see this in many of our videos. Condensation is also known as dehydration synthesis. So the two terms are interchangeable. You're still removing water. The big idea is you take the monomers or pieces and you bring them together. So let's start with your carbohydrates. So carbohydrates is the first macromolecule we're going to talk about. And some of the things that you learned about in the Quizlet vocab set is that carbohydrates are made of carbon, hydrogen, and oxygen in a 1-to-1 ratio. So carbon, hydrogen, and oxygen, and you count a 1-to-1 ratio. So if something is made of more than one carbon, for example, glucose has 6 carbons. So glucose is C6H12O6. You have other examples. You have other monomers for those carbohydrates. But typically what you're going to see, let's fix that mistake right there. Whoops. Typically what you're going to see is that 1-to-1 ratio. It may not be exact sometimes, but it's going to be pretty close. So that's H12O6. There we go. Carbohydrates are stored energy, and usually we call this fast energy. They transport stored energy within complex organisms, and structural molecules structural carbohydrates give organisms their shape. So when we talked about, or when you guys learned in the flashcards about different types of carbohydrates, we talked about glucose, which is your fast energy. We talked about structural carbohydrates. This is cellulose. And then we also talked about the function of recognition and signaling molecules that will trigger biological responses. And so the signals are on, or the carbohydrate chains are on the plasma membrane. And typically the most well-known example is going to be your blood type. So I'm just going to put A, B, O. Blood typing, those are carbohydrate chains, and we're going to talk more about those when we get to the unit on membranes. So when we're drawing these carbohydrates, there's a couple of things. We're going to dip a little into organic chemistry for this unit. And so I want to talk about what happens to carbohydrates when they're in an aqueous solution, when they're placed in water. Now, carbohydrate chains, when they're not in water, will form long chains, but when they're in water, they're going to form rings. And so we're going to practice drawing these rings because you're going to see these a lot throughout our course. First of all, carbons are the basis for organic life. And when we draw these carbons, we can simplify the structure of them. And here's how we do it. I'm going to draw a carbohydrate that has let's say that has 6 carbons. In fact, you know what? Let's go ahead and draw glucose. So glucose is C6H12O6. So when you draw molecules, and organic molecules, I should say, we typically represent carbons with corners. And so glucose is a 6-carbon molecule. It's going to have the shape of a hexagon. So I'm going to draw just a dot to get me started, and I'm going to draw a hexagon shape. So to do that, I'm going to draw another dot here, another dot here, another dot here, just to kind of give it balance. And so I'm going to connect these. So glucose is 6 carbons. 1, 2, 3, 4. I'm going to leave a space there. So this is carbon number 1, 2, 3, 4, 5. And so carbon number 6, let's move this out of the way. Carbon number 6 is actually coming off carbon number 4 there. Okay, here at this corner, in the case for glucose, this is an oxygen. So I'm going to draw a circle and I'm going to write an O to represent the oxygen that's there. And then I'm going to close this off. So this is a glucose molecule. Glucose is C6H12O6. And when you number these carbons, it's very important that you follow the same structure so that we can identify functional groups attached to these carbons. So if we number them, we're going to start from the first carbon off of the oxygen. So here's my oxygen, which means the first carbon I'm going to start at is right here. And I'm going to number them going clockwise. So this is carbon number 1, number 2, number 3, number 4. Right there is carbon number 5. And then this one up here is carbon number 6. So each dot represents, or each corner represents a carbon. And we number going clockwise starting from the first carbon right after that oxygen. Now those functional groups can be attached anywhere that carbon has free bonds or free electrons for pairing. So carbon can form 4 bonds. And this one, if you notice here, there's one bond here, there's one pair of shared electrons, and here's another one. So that means that carbon has space for 2 more bonds. And let's say, let's say that the first one I'm going to add is a hydroxyl group. Then I would draw just a line coming off of that carbon. And there's my hydroxyl group. And it still has room for 1 more because we're only using 1, 2, 3 of the bonds. So it's got space for 1 more. We're going to go ahead and just put a hydrogen group. So that's a quick little tutorial on how to draw these molecules. And what I need for you guys to understand is that the direction in which we are drawing makes a difference on the way that these molecules are going to behave. I want to give you an example. So at carbon number 4, there is another hydroxyl group and it's pointing downward. Okay, so try to picture in your mind what that's going to look like. Hopefully this is what you did. So carbon number 1, 2, 3 at carbon number 4, I'm going to draw the line going like this, not like this. I draw the line going downward and it has a hydroxyl group, so I would write OH. Now it may seem like it's not that big of a deal, but it's a very, very big deal. Because if the molecule has those hydroxyl groups pointing in a different direction, it changes the molecule completely. So now let's take a look at same molecule. I'm going to erase my functional groups here. Okay, now what I'm going to do is when I draw those groups, or when I draw those hydroxyl groups at carbon number 1 and 4, I can change the direction in which those hydroxyl groups are pointing. So now I can draw them pointing upwards, for example, like this. And that's a big difference. This right here is glucose, C6H12O6. I didn't draw all the H's and I didn't draw all the O's to keep it simple. The ones that you want to focus on are at carbon number 1 and carbon number 4. Those hydroxyl groups make a difference because this is glucose at C6H12O6. This one over here is still C6H12O6. 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 But because the hydroxyl groups at carbon number 1 and carbon number 4 are drawn pointing upward, see that? That hydroxyl group right there is pointing upward, whereas over here they're pointing down. Because of that difference, this is a completely different molecule. This is galactose. Completely different molecule. And when you combine galactose and glucose, you get lactose. So when these two combine by dehydration synthesis, you get milk sugar. But if you have two glucoses together, that's not milk sugar. That's something else. That's called maltose. So the direction in which we draw these functional groups makes a large difference in what that molecule is. Okay, so monosaccharides are the simple sugars. If it has 5 carbons, it's known as a pentose. So overall, you'll be looking for this shape. And remember, each corner represents a carbon. Now they can add additional carbons, for example, with ribose and deoxyribose, which are the sugars for genetic information. This molecule has a pentose shape, but it still has an oxygen group. So this is carbon number 1, number 2, number 3, number 4, and then carbon number 5 is coming off of carbon number 4. So that's still technically a pentose. Hexoses have 6 carbons in them. So they can be glucose, fructose, mannose, galactose, as we had mentioned earlier. Typically, these are going to be drawn as hexagons, but they may not necessarily be hexagons. Fructose is a good example of a molecule that's a hexose, but it's not in a hexagon shape. So this is what it's going to look like. You're going to have your oxygen group. It's going to have a pentagon shape. However, so here's carbon, 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 carbon. However, there's 2 additional carbon groups, one right here and one right here. So if you count them up, you get 1, 2, 3, 4, 5, 6 carbons, even though it's got that pentagon shape. So just make sure you count your carbons. Monosaccharides are covalently bonded by condensation reactions that form glycosidic linkages. So what you see here is the carbons, the 2 monomers, glucose and fructose, which come together to form sugar, like the kind of sugar you put on your Rice Krispies or your Frosted Flakes. And what you're going to see here is that OHH, right there, that OH and that H, or what we call H2O, is the removal of water, and they're showing you here. And then now what you see is this oxygen right here. Let's do that in pink. That oxygen right there is that oxygen right there. So that oxygen is now forming the glycosidic link between the 2 monomers. So it's this oxygen that's right here that remains behind and connects these 2 molecules together. That's your glycosidic link. Oh, and just to note, when there's 2 monomers, when there's 2 sugars in the carbohydrates, it's called a disaccharide. Di means 2. If it's more than 2, it's called a polysaccharide. Poly means many. And so there'll be many chains of those monosaccharides coming together. And one of the classic ones that we need for you guys to know is the difference between starch, glycogen, and cellulose. And the reason we need you guys to know this, because this is a great example. I'm going to put like a star here. This is a great example of how structure affects function in carbohydrates. So starches are members of the polysaccharide families. They're made of glucoses. Specifically, they're made of alpha glucose. And alpha glucose is just a type of glucose, as we saw over here, where the OH groups are both pointing down. That is alpha glucose. So starch is made of alpha glucose, as is glycogen. So starch is found in plants. Actually, we'll see this on the next slide. I won't write it. Starch is found in plants made of chains of alpha glucoses. Glycogen is also alpha glucose, but it's branched, it's highly branched. And so because the addition of the branches, it's going to behave differently. And glycogen is found in mammals. It's in your tissue, it's in your muscle, and it's in your liver. Cellulose is different. Cellulose is made of something that's called beta glucose. And so it has a different shape to it. It's got that hydroxyl group at carbon number 4 pointing up instead of pointing down the way it is in alpha glucose. So a real quick sketch of what that would look like. That's a terrible sketch. Let's try that again. A real quick sketch on what that would look like. There is my hexagon shape. Okay, it's glucose because I've got 1, 2, 3, 4, 5, 6 carbons. But for beta glucose, what happens is the carbon here at number 1, that hydroxyl group is pointing upward, whereas at carbon number 4, it's still pointing down. Remember that with alpha glucose, both of these were pointing down. Now in beta glucose, one of them's pointing up and one of them's pointing down. And it may not seem like such a big difference, but it's a huge difference. Cellulose is not digestible. You could eat all the you could eat all the paper in the world. You could eat grass, but your body can't digest it. We don't have the enzyme to break down the polysaccharides of beta glucose. We have the enzymes to break down starch and glycogen. That's not a problem. But cellulose is not digestible especially to mammals or to animals. Usually what happens is those organisms will form a symbiotic relationship with bacteria that break down the cellulose itself. So here is the images of the different polysaccharides. You have cellulose, which is very linear. And what they've done is they've just kind of drawn this generic hexagon shape to represent the polysaccharides. So they've simplified it even more. And so that's what this is here. You can see how the molecules are just this little hexagon shape, and they're unbranched. They're just long fibers that are bound together in those glycosidic linkages. This cellulose makes up the cell walls. And it's undigestible. It's really tough. Starch is branched. There's some branching involved, so it's not as compact as cellulose. And so starch is going to be stored as energy. Starch is the primary source of energy in plants. Potatoes have a lot of starch. Corn has a lot of starch in it. Then the highly branched form, which is glycogen, is, you can see all these, so not only are these long lines, but they also have branches like tree limbs coming off of these. And those contain additional carbohydrates, glucose. It's much more compact so that you can store it in your muscles and not weigh like 900 pounds. You can store it in your muscle, you can store it in your liver. And if you need this energy, you've got enzymes that can just break off the bits and pieces and then use that glucose for cellular respiration. Thank you guys so much for watching this video today. I hope it was helpful. Be sure to be on the lookout for the other videos on carbohydrates and on lipids, proteins, and nucleic acids. I'll see you guys later. Bye.